All right, welcome to another podcast. Of course, I am with the esteemed Dr. Jason Williams of the Williams Cancer Institute. Of course, part of the team as well. But we're going to be talking about a very much relevant in the news publication, actually research article that is super exciting in the arena of immuno-oncology. But Jason, you've been there. So it's almost like the party was developed by the Williams Cancer Institute, and it's just great to see others now joining the party.
Yeah. People are finally seeing, of course, seeing that injecting the immunotherapy in the tumors is the way to go, and obviously with the CD40, CD40 is an amazing immunotherapy agent. I call it the Swiss Army knife of immunotherapy. I mean, it does like a little bit of everything.
Yeah.
I mean, it's almost like, what doesn't it do? I mean, it really has like such a bridging point between, generating the initial immune response and carrying it over. It just has a lot of broad functions, and it's super important. So, I mean, this is a great agent, but one that has to be in the tumor because you give it systemically, you get all this off-target effect.
And of course, you can look at the show notes. The studies will be there. We encourage you to read them yourself. Don't take our word for it, but read them yourself. So I'm entitling this, Inject the Tumor, Ignite the Immune System. It's the power of intratumoral CD40 agonism. Go ahead. So, you kind of touched on it already, what CD40 is. It's kind of a master regulator. You called it the Swiss Army knife. I think that's actually a better term, Jason, because what's that, when you say Swiss Army knife, people have a picture.
Right.
When that comes to the immune system, what does that mean?
I mean, it's like it does a lot of things. Maybe it's not perfect on everything, but it makes a lot of things happen, and you can get a lot done with one tool, right? Whereas like other things are very specific, whereas this one has just so many things that it does. It's just kind of incredible. And that's why I think that Swiss Army knife seems like a good analogy.
Well, I think that's a better one than master regulator, because when we talk about the master regulator Swiss Army knife, it's because it's having multiple immune functions. I mean, it's priming the T cells, it's enhancing antigen presentation, it's really bridging the innate and the adaptive or humoral immunity. So now CD40, that's a drug, but it's a component of the immune system.
Yes, it's a receptor, and this is a CD40 agonist, so we're using a monoclonal antibody that binds to that receptor and stimulates the CD40, and so that's its role. And but it's amazing, all the things that's involved in, because you get activation of that receptor, and it turns that some of the innate immune response into a more adaptive immune response, and then also you've got even going on to recruitment of like natural killer cells and dendritic cells, all this other type of things with CD40 is just it's so multi-purpose. So.
I mean, what you're describing there is the pieces of the puzzle, the team, if you will, that comes together that gives us the abscopal effect.
Yeah, I mean, for us, and that's what, like when I work with our therapy, like our CT therapy, we have CD40 and that, and and CD40 is like your base. I mean, that's the one where you just have to have it. And if you only got one thing, that's what you got. But you need other things to make it work better. But that definitely is like the real linchpin. That's like the key component.
So I want to go through a timeline, because we'll get into this particular study where they only used a CD40 monoclonal antibody, because I want to insert you and the work that you pioneered at the Williams Cancer Institute into this timeline, because in the 1990s, CD40 was first discovered as a master regulator or a major contributor to Swiss Army knife to antigen-presenting cells. But in early 2000s, what we saw is that initially it was really activating and enhancing the innate immunity, cytotoxic T lymphocytes, antigen presentation, leading to tumor regression in mice. And then you've mentioned Van der Heide quite a bit. Elaborate on that, because that really brought it to the public consciousness in the scientific arena.
Ron Hudd at UPenn, did a lot of studies looking at pancreatic cancer, and he was doing systemic CD40 with with combination of chemotherapy, and they got some really amazing results. The problem is the systemic delivery has an added toxicity, because CD40 is great for activating the immune response, but if you're giving it systemically, then you're going everywhere, right? And so you have this off-target problem. So you need to be able to put it at the tumor site. That's where, that's the difference. And, there's a lot of these immunotherapy drugs that are really, can they work systemically? Yes. But are they more powerful in the tumor? Most of them are more powerful in the tumor. I mean, that's where you need to change the environment. So you'd rather put it more there. You did it from the inside out instead of the outside in. So that's, but yeah, it's a very, very amazing CD40. And
a colleague of mine who I respect immensely, I don't think you've had a chance to meet her, I'd love to connect you with her, she's a breast surgeon, but she actually said, don't focus on the tumor, focus on the root causes. Now, I would say, well, there's some truth to that, and there's some not truth to that, because when patients come to us with stage 3, stage 4 cancer, hopefully stage 1, stage 2, where it makes the immune system's job much easier, there's a big fire.
Yeah.
And if you just say, well, let's go looking for the faulty wires that caused the fire, the house is going to burn down.
Exactly. Yeah. That's the same thing with cancer. I mean, you can't expect to go and fix that wire problem and then you're going to extinguish the whole fire. It's a whole other situation. I mean, cancer, once it's kind of taken over, I mean, there is maybe the root cause, but you really have to address the overall picture and what's going on. So yeah.
But the big advantage here, the big benefit is because you're avoiding that systemic tumor approach. You're going to the tumor to activate the immune system, harness it. We'll talk about how that occurs in a second, but then that gets transferred systemically.
Yeah.
So it's a much safer, you said, avoiding those off-target toxicities.
Yeah. The thing is that you're affecting those immune cells that are in that environment around the cancer. That's what you want to affect. You don't want to affect immune cells that are elsewhere, that are near normal cells. Right. You want to affect the ones that are around the cancer. That's the ones that you want to change, and that's why you have to put the drugs at the cancer site.
So basically they started to discover, because actually the first phase 1 trial in 2008 was published, following his work, and they found actually some regression of tumors.
Yeah.
But as you mentioned, the systemic toxicities were just too great.
Yeah. The problem is that when you deliver these drugs intravenously, huge amounts going to hit other areas, particularly a lot of it goes in the liver. And then, that will cause the immune system to want to attack the liver. You get this a real problem with autoimmune hepatitis, I think, is a big issue with CD40. And so, that's why you need to be specific in your area. You need to try to get the immune system to attack the cancer, not all over the body.
So then they started, as we go through the 2000 teens, if you will, they recognized the challenge of the systemic delivery. And so they thought, well, you know what, let's try to provide a better binding molecule and thus be able to reduce the dose, but then still give it systemically IV.
Right, yeah.
And they ended up with the same problem.
Yeah, they try to get it more specific, try to get it to go more to the tumor site, and that's a reasonable consideration. But again, the thing is that we can put a needle in the tumor. We do this for a biopsy, so certainly we can inject the medications in there, so we might as well just do it that way. That's just much simpler than trying to figure out a way to hone it in. That's much more complicated. It doesn't work as well.
So what they did is 2017, 2018, that's where then this concept of, okay, maybe it's not that we need a better binding and lower dose, we just need to change where we deliver it.
Yeah.
So 2017, 2018, that's really where the paradigm shift happened. There was actually a publication, get a check out the show notes, you see it there. When did the Williams Cancer Institute start bringing this into the clinical fold for patients?
Yeah, that's a good question. I mean, I definitely say, probably say it's been at least four years, maybe could be 5 years. We had looked at it, we wanted to use it, we were using a bunch of other agents, and then once that one kind of came on board, that was a game changer for us. We particularly cancers that we were not seeing great response in, like pancreatic cancer, respond very well to CD40. I mean, it really turned things around a lot, and so it really felt that ended up being the real key component.
So if you got stranded on a remote desert island,
Yeah.
would this be one of the three that you would take with you?
This would be the CD40 is your number one. Yeah. So.
And like you said, it's a part of the ongoing phase 2 SYNC-T trial.
Yeah. So obviously we have a combination with the other drugs, with CD40 certainly being the key base.
So basically then, when this really started to hit phase 1, 2019, 2022, that's basically about the same time that was being incorporated into the protocol or the treatment strategy, better statement there, I think, at the Wings Cancer Institute.
Yeah, because so we when we designed our SYNC-T drug, we looked at the ones that what are the agents that we thought that were really good, and we had various ones that we were looking at, but yeah, at that point CD40 would so the thing is that maybe it was even it could have been longer. I'm trying to think how many years ago that was when we were like going through that process of deciding that was the drug that we wanted to use, but we're seeing that seemed to be the one. I mean, playing around with others, we tried, like, they've got the costimulatory ones, you got OX40, you got 4-1BB, things like that. And you got, like, CTLA4 is always it's a pretty good base as well. I mean, CTLA4, it's probably number 2 to CD40, I would say. But then you have to have the two together. I mean, it's the thing is you need it all together. It's like you can't have just one part of it.
And we'll touch on that at the end here. So basically, looking at CD4 agonism, it's really working on dendritic cell activation. It's working on T cells. It's working on B cells. It's working on macrophage activation, particularly in one polarization. Because I think a lot of people get stuck, Jason, into this mindset of the magic bullet theory. One drug to do one thing, but CD40, it's like a pebble in a pool of water early in the morning, rippling through the immune system.
But there's no one drug, and you got to have that concert of things going on. The immune system, to get the appropriate signal, is not going to be just not going to be just PD1. It's not going to be just CTLA4. I mean, even CD40 by itself, not quite enough. You need all of these things working in concert together, and the immune system sees it that way. It's a code. It's I mean, that's like if like the safe. I mean, if you want to open it with one number, right? You got to have all of them all together in the right sequence.
So you're describing what you're doing is conducting a symphony with these different therapeutics that are working in the immune system. So I guess we can call you Dr. Williams. Obviously we do, but it's Maestro Williams, right? Because you're conducting a symphony with the immune system.
It is like an art to it, and the thing was that it's interesting to start experiencing seeing in human patients, because like you always had you had the mice and you had the individual stuff and you try to put that together, but then as you start seeing the humans and then you start seeing all the different things kind of working together and start getting a feel for it, really that changed things for us.
So, people go, well, why does this not work on everybody? What we're starting to understand is obviously cancer is not a solid ball of cells. There's actually this tumor microenvironment. There's different components within it. But we'll talk about it because in this study what they found is there are these substances called tertiary lymphoid structures, which are kind of ectopic lymph nodes. And what these are doing is it's basically setting the stage for a training camp right nearby the tumor for these T cells to be primed. And this is what is really eliciting this systemic immune response in a predictable manner. And CD40 in many ways is driving this to a degree, and that's what this study showed. These tertiary lymphoid structures are kind of like your military base. I mean, if you're going to be attacking an area, you want to have your supplies and all your stuff close by, and that's why these tertiary lymphoid structures become toward that, where the immune cells can gather and sort of unite to try to fight the cancer. So we get that. But you mentioned the combination, because one of the things that the conclusion of this study they mentioned was they said, well, now what we need to do is to figure out what combination of therapies works with. And that's where, it's like, well, sorry, you've already figured that out.
There's we have certain regimens, certain cancer types, we say, okay, look, because again, in the perfect world, what would you do? You would start with one set of drugs and you would start priming that immune response, and that would probably include CD40 and CpG and CTLA4 for sure, coming in there. And then as the immune response is developing, then you would want to try to push and recruit that immune response. So then you start looking at like co-stimulatory things, and you got like 4-1BB and OX40, things like that. Then you arguably there's some cytokines that you could use as well. There's various ways that you could try to push that response. And then of course, like the other side is you got these breaks, where the cancer is going to try to start shutting off the immune responses. With the PD-1, the LAG3, there's ICOS, there's VISTA, there's TIM3, there's all these off switches that, so then it's going to start hitting those. And so, in a perfect world, you would adjust and try to do it. So, we have a base that we know for certain cancers it works and it just carries through. Other cancers are more complicated, and other cancers you need to be able to be adaptive and say, okay, we're going to start with this, we're going to change this, we pivot, and now we're going to start loading heavy. And as you get pushing the immune response button, you got to get heavy on all the checkpoint blockers. You got to take all that down, because otherwise the cancer's going to shut it off.
So it's complex.
Yeah.
Now, one therapy that we do that they didn't have in this study, obviously, they just injected intratumoral CD40, is PEF. And PEF generates tertiary lymphoid structures.
Yeah. So obviously you get the immune stimulation of PEF. You get the tumor killing, and it just basically functions like an immunotherapy agent itself. Even though it's a procedure device, and killing the tumor with electrical shock, and that. But it's amazing how that creates a particular immune stimulation. So it's an added bonus. Can you achieve it without it? Yes. Okay, you could certainly do it without, but adding it to it usually gives you some benefit.
Yeah. So basically, the issues you described was the systemic toxicity, off-target toxicity. You mentioned the hepatotoxicity, really being driven by the cytokine release syndrome, but also really bad thrombocytopenia. So the challenge that they had was this: we've got a therapy that seems to work.
Yeah.
So Swiss Army knife. Yet the toxicity is so great that they couldn't figure out what to do. It took some time. But you were already in the process of figuring it out, because going back to your residency and fellowships, you knew the tumor was the means to activate the immune system.
Yeah.
Or at least that process was beginning to percolate.
Yeah, I mean, it's like you imagine it's like it's like a war going on there, and there you got like your enemy is located there, like you need to go where the enemy's at, right? I mean, that makes the most sense. I mean, where the highest concentration of the enemy is where you should like focus your attack, and that's how it should be in cancer.
Because that tumor burden, that plays a big role, right? In terms of treatment efficacy, activation of the immune system. Because you have to recognize the fire in front of you, the emergent issue. We can focus on the root causes simultaneously, but you said with that tumor burden right there, you got to go get it.
Yeah.
You got to do something about it.
Yeah, I mean, basically, what it seems to be is that, what I kind of say is that whoever controls the immune system being the master constructor and architect of the body, controls the body.
Yeah.
And so when the immune system's working with the body, that's one thing. If cancer, though, hijacks it, and it starts taking over, now it controls the immune system, now it's working with the cancer, and that's what cancer does. I mean, it has to basically convince your immune system to work with it. So that's really, it's a very, very sneaky how cancer does that, but it understands it.
So cancer will recruit the immune system to itself. It'll then turn the immune system on the immune system to protect it.
It will start getting the immune system to work for it, because it really, cancer cannot spread and do things alone. It has to have help, and the immune system is what helps it. And so for cancer to go to other places, it's not just the fact that the cancer just lodges and ends up there. Maybe that can happen, right? But usually the immune system has to have helped it to get in there, and so that's where that was the interesting thing. And to me, when I saw this in the research, like, so Case Western this special microscope, and they were they're trying to look at like, you know, cell migration and stuff. And I can just remember, I wasn't involved directly in the study, but I was, you know, involved with these like graduate students and researchers, and they said, "Well, let me tell you something really interesting. You know, we see this breast cancer, but what we're seeing is that when it goes to the bone, the immune cells are going to the bone first, and they're normal, and they're prepping the bone, and then the cancer comes behind it." And I was like, "Ooh, okay, wow, that well, obviously makes sense now." But you know, I don't think people really think about that. I think we were just thinking that, you know, you got bone, you got liver, wherever cancer just lodges in there and starts growing, right? But that the immune system is actually prepping these areas for the cancer to go there.
You know, it's funny you mentioned that, Jason, because there is a theory called the seed and soil theory, where in the distant metastatic sites, what happens is these, what they call pre-metastatic niches, the soil is prepped.
Yeah.
So that then when the seed, the circulating tumor cell, arrives, and the immune system appears to be that soil preparation.
It is. Yeah.
But it's not doing it, it's being told to do it.
That's right. Yeah.
It's being told to do it.
Yeah. So then these are normal cells, right? They're not cancerous. They're your normal immune system. But now the cancer is controlling it. You know, once the cancer starts controlling your immune system, well, then you start having problems.